Welcome everyone to the inaugural Alan J. Magill Malaria Eradication Symposium. It's a full room. I think it's going to be a full room, but it's not quite full. There are still seats in the front, so please grab a cookie and a drink and come on up to the front. We've got a great program for you. My name is Plowe from the University of Maryland Institute for Global Health, and I'm co-chairing with Regina Rabinovich from the Harvard School of Public Health. Just before we begin, if we could have that one slide, please. I wanted to re-announce the Alan J. Magill Fellowship and Forums. So Steve Higgs last night during the plenary session mentioned this wonderful partnership between ASTMH and the Bill and Melinda Gates Foundation that in the memory of Alan, our dear friend and colleague who we were grieving just a year ago at last year's meeting after his tragic death, in his memory and to fulfill his vision and his legacy of the dream of eradicating malaria, the foundation has made a 20-year commitment to support this symposium. This is the first inaugural symposium, a forum that will focus somewhat less on the science that we'll talk about here and more on kind of policy and advocacy in Washington around the time of World Malaria Day each year, as well as the fellowship that Steve Higgs described to promote career development for scientists, program people working on malaria eradication in less developed countries. So with that, I would like to ask that we begin the symposium with an introductory video from Melinda Gates. If we can pull up the video, please.
Last year, the world lost a great leader in the fight against malaria. Dr. Alan Magill was a brilliant scientist who understood the realities of public health practice. When Bill and I talk about the kinds of leaders needed in global health, we simply say we need more people like Alan Magill. Alan was passionate about the injustice of malaria, wiped out in some of the world, but taking such a terrible toll on the rest. This appalling inequity demands all of us to keep working until we finally eradicate malaria. There is so much progress to build on. Since 2000, malaria death rates have fallen 60% among at-risk populations. 5 countries have been certified malaria-free, and 3 more have begun the process. To continue this momentum, countries and regions must have the flexibility to design interventions based on their own needs and objectives. I know many of you are doing great work to create better tools and methods for malaria prevention, diagnosis, and treatment. Of course, research and development will continue to be important. That's why I'm proud of our foundation's new partnership with ASTMH that will honor Alan's memory and advance the cause he championed. I look forward to the new forums and fellowships both strengthening collaboration and, most of all, inspiring a new generation of young scientists. I am sure it will help us find more people like Alan Magill. Thank you.
I look across the room and it's great to see so many faces I know and so many young people that I don't know. So it's the right mix of people. And I'm delighted to launch this by asking Bruno Moonen to join us up here. He's going to speak on the case for malaria eradication and the challenges I think that we will be facing. Please join us.
Good afternoon, and thank you very much for all showing up. It's wonderful to see so many people at this first Malaria Eradication Symposium. So I had a title that was a little bit provocative on purpose, that said that malaria eradication is both necessary and achievable. But of course, when you think about necessity, there are people who have defined that in different ways than we sometimes think about this. So let me first take a step back and say, we tried before with the Global Malaria Eradication Program to eradicate malaria. But did we really try and did we really fail is maybe one of the questions that we need to ask ourselves. And this is a graph from a paper or even a small book that was published by Pepe Najera, who pointed out that Sub-Saharan Africa was never really part of the global malaria eradication program. And despite being portrayed as a failure, there was a substantial reduction in malaria in many malaria endemic countries, and even some countries actually fully eliminated malaria. But what happened? Well, what happened was that the program was probably a little bit overly optimistically counting on one single tool, and didn't take into account the differences in the epidemiological setting and context. And they were not flexible enough to address some of the challenges that they faced. When then, of course, they were faced with the challenge of both drug and insecticide resistance, they didn't have a real plan B. What happened then was that the donors withdrew their funding to the special account, the WHO special account, and the program actually came almost completely to a standstill. Pepe Najera pointed something very important out to me when I looked at this graph. He said, if you look at the contributions to the WHO special account, initially you see that the US is of course the largest contributor to this program, and they stopped in the late 60s contributing to this program, after which there was really almost no funding going into this program. So why is there a spike all of a sudden of, in this case, European contributions? Well, it was actually because it was a reaction to a resurgence of malaria, and in some cases quite devastating epidemics. What also happened was that I think people became less ambitious. And what we're seeing is that you had some elimination during the global malaria eradication program and even before, a number of countries afterwards, but then really a valley of death, where there was no elimination at all. And it was only in the early 2000s that countries started to think again about aiming for malaria elimination. So the question is then, will history repeat itself? Well, we have multi-drug resistance, and we have insecticide resistance. So you could argue we are exactly at the same point as where the global malaria eradication was at the end of the 60s, the beginning of the 70s. Is the money still available? Well, we're seeing money plateauing a little bit, and it remains to be seen if the commitments from the major donors, including, and very importantly, of course, the US government, will be maintained. And it's clear that for the malaria eradication, to achieve malaria eradication, that we will need more resources today than the ones that we have had so far. And the question then is, do we really want to relive the 70s and the 80s? So on the bottom you see, of course, a number of countries where malaria kept going down, but largely in places where there was substantial socioeconomic development. And in Africa, it wasn't great. And I know there are many people in the room here who were working on malaria in the 80s and the 90s, and who remember a time where they didn't have effective drugs, they didn't have very good prevention measures, and they didn't have any funding. And it was a terrible time for malaria in sub-Saharan Africa. And I don't think that that's something that we would want to see come back if we are not addressing drug resistance, insecticide resistance, and if the funding goes away. So if we don't want to relive the 70s and the 80s, there might be other reasons why we don't want to do that. But then the question is eradication really an extravagance? And of course, for us the answer is no, because we know that if we don't aim for eradication, we will see this problem come back over and over and over again. And we will need to continue developing new drugs and new vector control tools, because resistance will appear, and a parasite that has lived with us for millions and millions of years will continue to adapt when we try to fight it with the tools that we have today. Our vision, of course, is quite clear. The Bill and Melinda Gates Foundation and our co-chairs, Bill and Melinda, have said that we should aim for malaria eradication. And I would like to point out that this is very much an equity statement. This is not some kind of ambitious goal because we think it's a good idea and we like to innovate. It's an equity statement. What Melinda said at the forum was, if you don't eradicate malaria, where are you going to stop? What are the countries that we would pick today where we would say, it's fine, we're just going to do control in your country, we shouldn't aim for elimination, let's stop at one point. And that is just not acceptable. Is it achievable? I always like using quotes because you can both use them in your advantage and they can be used against you. So this one works in my advantage, this one probably not. So there are, of course, some reasons for optimism. And I'll go through them a little bit step by step. The first one is that we don't think that to achieve malaria eradication, this is going to be a global kind of Texas all-in campaign. We think there are some helpful global trends in the background. And the good thing about accepting the fact that we believe that it's impossible to do it with current tools is a good thing, because we believe that that spurs innovation and will continue for people to, and many of you here in the room, to invest in their careers in research and development. We have clearly acknowledged that there is a problem today with drug resistance and insecticide resistance. And what's also very important is that this is no longer a top-down kind of pushed approach of global eradication. Countries and regions want to eliminate malaria, and we should take advantage of that momentum. So what are we talking about? This is Emilio Pampana's definition of malaria eradication. It's removing parasites from people until there are no more parasites left to be transmitted. And this is the definition that I think every year until 2040 at this symposium should be put forward, because this was Alan McGill's definition of malaria eradication. So first of all, true eradication and not an all or nothing campaign. So when I say true eradication, I don't want to mention the infectious diseases for which I don't think it's the case, but I believe that for malaria, when we eradicate malaria and when there is no more parasites left, that we can actually stop with malaria interventions. We might still need some integrated vector management, we might still need some integrated surveillance, but I believe that there should be no more vertically implemented malaria interventions at that time. I don't think that's the case for all infectious diseases where people are aiming for eradication or thinking about it. And unlike polio, where today every single country is still vaccinating birth cohorts until polio is globally achieved, what we see in malaria is that many places that are today malaria-free and that were malaria endemic a long time ago, they don't have malaria control programs anymore. So it is not this global all-in campaign that you see for other infectious diseases. We believe that if it's not a global all-in campaign, what is it? Well, I think if we address this problem region by region, and this does not mean that this is not a country-by-country effort, it's just that when a region completely eliminates malaria, it is very likely that it will be difficult to reintroduce malaria into that region. A good example is, for example, Latin America. In Latin America, they have not really seen chloroquine resistance that was quite prevalent in South America. So it seems that those parasite populations are not really connected or linked to each other. That also means that reimportation from South America into Latin America is probably something that will be minimal and manageable. So if you can eliminate in that region, then you will probably be able to maintain it. And people have pointed out in some scientific publications that malaria is probably a sticky state, which is going to help us when we try to eradicate. In the background, we have a couple of very helpful trends. We see increasing urbanization, not necessarily a good thing for many infectious diseases. And we know that there is urban malaria, but largely urbanization helps us because we see less malaria in urban areas. And we have a reduction in poverty, and malaria is a disease of the rural poor. So if you see increases in urbanization, so less rural people, and if you see a decrease in poverty, that's going to help us to eradicate malaria. As I said before, an ambitious goal spurs innovation. A good example is the Malaria publication that is actually being refreshed as we speak. that is really thinking hard about what needs to get done to achieve malaria eradication. And we already see some programmatic innovations where people are using high-resolution mapping. We're seeing people using mobile phone data to assess mobility. We're seeing mobile phone data being used for reporting. We already have some new products. We now have a highly sensitive rapid diagnostic test, 10 times more sensitive than the current RDTs. And we have very soon, we'll see new combination bed nets. And the R&D pipeline is quite strong. We have work going on vaccines, drugs, vector control, and diagnostics. We are reacting to drug and insecticide resistance. WHO has issued very clear guidelines on how this should be done, and even our R&D efforts always take into account that whatever we develop needs to be able to counter both drug-resistant parasites or drug-resistant vectors. The regions are very ambitious. You can have long debates on whether or not a 2030 target in Africa is the right target, but you cannot deny that they are ambitious and that there is clear demand for elimination and more ambitious goals. So what do we think malaria eradication in the 21st century is going to look like? And more in specific, because I don't think that this goes for every single infectious disease, malaria eradication. So we believe that this is not a global effort. This is going to be a regional effort driven by the countries themselves. It's about context-adapted implementation and sustained R&D, which is going to be critical. And it's a move from vertical programs to horizontal programs where we move from malaria-specific vector control and malaria-specific treatment and diagnosis and surveillance to integrated surveillance and integrated vector management. Thank you.
So we're going to go through these talks and then bring the whole panel together because otherwise we will never get to the end of the session. And I think there is a really wonderful opportunity for interchange. The next speaker is Dr. Pedro Alonso, Director of the Global Malaria Program at the World Health Organization, talking maybe about historical lessons and new strategies, but he's always a surprise, so we shall see.
Well, thank you very much. And first of all, thank you very much for the opportunity to be speaking at this symposium that remembers our good friend and very much missed colleague, Alan Magill. So, as Bruno was speaking, I was getting slightly worried because these days we seem to have pre-breakfast meetings every morning at 7 in the morning, solidly, and as you will see from my talk, there seems to be not just that we share slides, but thoughts too. And so please bear with me on this. So a few comments on the past and where we're maybe going in the future. So as Bruno has alluded to, in 1955, the World Health Assembly that came together in Mexico City on that year launched the first global malaria eradication program, and the then Director General of WHO, Marcelino Candao, said, "We have the tools, we know what needs to be done, and it is simply a matter of going out to do it." Well, it's always complicated to do this sort of historical revisionism and how wrong was he or how right he was. As Bruno was saying, they genuinely thought they had the tools. There was still highly effective DDT with its residual effect. There was chloroquine, safe, inexpensive, and highly effective. DDT, which the first signs of resistance had been identified a couple of years before that in Greece, as I said, was still highly effective and indeed did prompt a strong rationale for an accelerated effort towards elimination. So they did believe they did have the tools. They thought what needed to be done, and that's probably where they were most wrong. And as again Bruno has alluded to start with, they didn't know what to do in the highly endemic countries of Africa. And that's one of the reasons that not much actually happened in Africa during the eradication campaign. And and the last sentence, that it was simply a matter of going out to do it, reflects what was perceived as this was an operational effort that needed to be done. It was a managerial one. Malaria was not seen as a problem to be solved. It was seen simply as a task to be performed. And that probably was not the right approach. 15, 14 years went by, the decrease in the funding into the special account, the lack of progress in Africa, where not much was attempted, the growing problem of both drug and insecticide resistance. Soon, probably by 1963 and 64, there was a recognition that this was not going to be achieved. And in the World Health Assembly in 1969, the campaign, the program, was brought to a halt. But I'd like to draw your attention on the actual wording contained in the World Health Assembly resolution of 1969, the one that is credited for stopping the eradication effort. And they said, quote, that complete eradication of malaria from the world remains a primary task of national public health organizations, and that even in the regions where eradication does not yet seem feasible, control of malaria with the means available should be encouraged and may be regarded as a necessary and valid step towards the ultimate goal of eradication. In other words, the World Health Assembly never gave up on the goal of eradication, and actually visualized in a very explicit way that control, reducing disease and death, was actually on the path to eradication. And I think it's really important to retain this quote from the 69 Assembly, because it remains hugely important for us today. The slides we also share these days, Bruno and myself, what was achieved in the 14 years of the eradication. Indeed, 15 countries and 1 territory achieved malaria elimination and were indeed certified. In the tail of the eradication campaign, a further 7 countries and 1 territory did eliminate malaria and got certified. And then we entered this long 20-year-old period where nothing happened. And as we know, malaria came back with a vengeance. Mortality and morbidity increased, and it took a huge effort to start reversing that. And the period 2007 to 2016, from the elimination perspective, starts witnessing this comeback. We used to say, even Melinda on her video says 5 countries. It's actually 7 countries, and admittedly she probably taped the video a month ago, because in the last month 2 further countries have been certified. The first one, and one which brings remarkable emotional notes to all of us, is Sri Lanka. Sri Lanka is always mentioned as the case of a country that nearly achieved elimination. There were only 12 or 18 cases left, and the effort was stopped just before achieving success, and malaria came back again with a vengeance. 15 years ago, Sri Lanka had over 200,000 cases, and within just over a decade, they've managed to reach 0, sustain 0, and be certified by the Director General of WHO last September. In the last week, I believe, or 10 days, Kyrgyzstan has also been certified malaria-free. And so this reflects this remarkable progress that we've achieved over the last 15 years, what we often refer to as the unprecedented progress, with a 60% decrease in mortality globally in the at-risk population and 37% decrease in case incidence. And this has happened globally. This is the same figures for the different WHO regions. So the progress has not been in just one place, it's been throughout the malaria endemic world. And please also note the orange line to the left, the one that reaches 0, is for the first time last year, an entire WHO region achieved 0 malaria indigenous cases, the European region. And you would say, well, you know, the European region, that was to be expected. Well, actually, the Euro region contains all the ex-Soviet Central Asian republics, that following the collapse of the Soviet Union saw a massive resurgence in malaria, and they have managed to reverse it, drive it down, and reach 0 cases, and the last countries are actually being certified. For the first time, an entire region is malaria-free. And not only has disease and death come down, but the number of countries with just a few cases has been increasing. The different blue graded lines represent the number of countries with less than 1,000 cases per year, less than 100 cases, and less than 10 cases. So a steady increase over the last 15 years in the number of countries with few malaria cases. In the face of this progress, last year, the World Health Assembly endorsed a new global technical strategy, the first strategy developed and endorsed by countries since the 1993 strategy on malaria control, which sets these goals, these milestones, and these ambitious targets of reducing disease and death by 2030 by over 90% and achieving elimination in at least 35 countries, under the vision of a world free of malaria. And again, this is significant. It's the third World Health Assembly resolution that speaks of a malaria-free world. And I will be coming back to this again. Beyond that, over the last few months, we've been looking at those countries with few malaria cases and trying to understand, both in projecting on the grounds of the decreasing slope of the last 10 years, projecting them forward, plus an assessment of operational feasibility and capacity by the country, which countries could actually achieve 0 over the next 5 years. These are the countries with less than 100 cases. These are the ones with less than 1,000 cases, and these are the number of countries with less than 10,000 cases. And essentially, we published 3 or 4 months ago a report which suggests that actually 21 new countries could achieve 0 cases by 2020, which is only 4 years ago. A sign of the growing momentum, as Bruno was saying, that countries and regions want to eliminate malaria and are moving forward in a very decisive way. The interesting thing about this list of 21 is that it actually speaks to all regions. It's not just the Americas, where probably the biggest contribution is going to take place. The Eastern Mediterranean region, including Iran and Saudi Arabia, Southeast Asia region, the Western Pacific, but also the African region. And here there are some, one would call some low-hanging fruits. Algeria, just 6 cases, and probably we're actually investigating that literally as we speak. Probably all imports. So Algeria may have actually already eliminated malaria. Botswana, very close. Cape Verde, we're talking of less than a handful of cases. Comoros. South Africa is going to be a challenge, but we believe it can be done, and the country has pledged to do it as well as Swaziland. So a large movement of countries throughout the world, throughout all the regions, aiming for elimination. Now, let me quickly move over to a few more generic principles that I believe we need to retain in this discussion. A lot of them inspired by Walter Doodle and his book on the principles of disease elimination and eradication. I believe they remain relevant to all of us. 3 indicators of primary importance as to whether a disease is eradicable or not. A, effective interventions are available to interrupt transmission of the agent. Are we there in malaria? Well, the fact is that a good number of countries, as we've just seen, have actually eliminated malaria with currently available tools. So the answer is probably yes in a good number of places, perhaps not sufficient to achieve elimination in the hardest places, and therefore a renewed call for the essential work in R&D that needs to happen all along the process until eradication is achieved. The second principle is, are there practical diagnostic tools with sufficient sensitivity and specificity to detect levels of infections that can lead to transmission? Well, we actually have a problem still in terms of defining at what parasite densities is transmission and how effective is it, but I'm sure a lot of work is going on there, and indeed new diagnostic tools are being developed, highly sensitive ones that may play a useful role. But once again, on the face of light microscopy and RDTs, elimination has been achieved in a number of places. And finally, humans are the essential for the life cycle of the agent, and there are no vertebrate reservoirs that do not amplify in the environment. That is true for 4 of the 5 species. Pinot-Lazy is a different it's a different issue. So I think on some of the key indicators we are on reasonably good grounds, but no time for content and more work needs to be done. Economic considerations that should be brought to the table. I think these are really important at the broader policy discussions, particularly thinking on the resources for elimination or eradication, and whether they should be used for this or for other non-health projects, alternative health interventions, continued control of the condition, or the eradication of other eradicable diseases or conditions. In other words, malaria does not live in isolation from a complex world with relatively finite resources where decisions need to be taken. Third category of social and political criteria. Malaria, in this case, should be a recognized public health recognized of public health importance with broad international appeal and be perceived as a worthy goal by all levels of society. And I think, in that sense, we are in not too bad ground. Secondly, a technically feasible intervention and eradication strategy. Again, countries have managed to eliminate malaria. A good number of them will be eliminating malaria in the next few years. Have we got all the strategies to address elimination in the hardest places? Probably not, and not because we don't have the tools or the strategies to achieve that. Thirdly, we must carve a broad international consensus. And I think we are on our way, but a lot more work needs to be happen. Consensus on the priority and justification for the disease. And this must be developed by technical experts, the decision makers, and the scientific community. And often our community is very good, because it's a lively and vibrant one, to engage in complex and at times futile discussions. Fourth, political commitment. And political commitment must be gained at the highest levels, following informed discussion at regional and local levels, and a clear commitment of resources from international sources is essential from the start. And I would add, of domestic resources. A resolution by the World Health Assembly is probably a vital booster. Now, on the political commitment, I think we often see, as Bruno mentioned, high-level declarations from high-level officials. But as we will be reporting on our World Malaria Report this year, domestic funding, Bruno alluded to overall funding for malaria has probably plateaued. What to me is a much more worrying fact that we are documenting this year is that domestic funding has remained stagnant over the last 10 years. And unless political commitment by the affected countries translates into domestic funding from the affected countries themselves that reflects true ownership of the elimination effort by the country itself, we won't be able to eradicate malaria. And we need an advocacy plan, and the eradication program must address the issues of equity and and be supportive of broader goals that have a positive impact on the health infrastructure. To address some of these issues, WHO, with the support of partners, has established a strategic advisory group on malaria eradication at the request convened by the Global Malaria Program at the request of the Director General of WHO. The first meeting of this group together with some of the WHO collaborating centers and key partners, took place on August the 28th, 2016. And broadly, this committee aims to analyze what future scenarios for malaria may look like, taking into consideration a broad set of biological, technical, socio-economic, political and environmental determinants, including potential products of innovation. We are all fully aware that the progress in the fight against malaria has not just been because of bed nets and ACTs. Economic and social development plays a critical role. The role of urbanization, electrification, mobility, many of those factors play on our favor in reducing malaria, but others do not. Having a broad understanding of those determinants, their potential impact in the coming years, and trying to understand where may we be by 2030, which if the GTS says we will be left with still 20 million malaria cases and somewhere around 20,000 deaths, will help us see if and when a further effort to launch an endgame may be reasonable. I won't dwell on malaria definitions, elimination and eradication, because this is an expert community, but believe me, there are still significant confusion out there around this. Among other things, because definitions do change. They were not the same as the time of the Global Malaria Eradication Program than they are now. What we now call elimination was then called eradication. And I think the current WHO terminology helps to sort that out. But in the face of SDG goals, ending malaria epidemics, and what does that actually mean, and how that does not equate to eradication, this clarification is often useful. From our strategic advisory group, a few initial thoughts that are emerging and that may be worth sharing with you, and many of them, again, have aligned with some of the comments of Bruno. The first one, which I think is important because it's a document that will be going forward to the Executive Board of the World Health Organization, is reaffirming the unequivocal commitment of the World Health Organization to malaria eradication. And this is not because me or any other member of the WHO says so, but because there have been by now 3 World Health Assembly resolutions, the 1955, the 1969, and the 2015, that equate eradication and the vision of a malaria-free world. A paper in that sense, as I say, is moving forward in our governing bodies for reaffirming this unequivocal role. There is no discussion, is WHO in favor or against eradication? We are unequivocally for eradication because that's what our strategy, as approved last year, calls for. Secondly, differentiating what at times is one of those unhelpful false dichotomies. Is eradication feasible? Well, from a biological point of view, there is no doubt. I'm sure everybody in this audience would agree to that. Is it operationally feasible? That's a different question. And that may be different in different parts of the world. So just using the term feasibility to relate to eradication is probably not a useful one, and we need to start separating one from the other. The third one is the recognition that we are moving towards eradication. We are moving towards a malaria free world. We're not in a campaign to eradicate malaria, but we are moving towards eradication. And probably we should use just the word campaign, if we ever use it, to refer to the end game. Even when it is within reach and there's a specific time date where we believe it can be achieved and there is full funding and full technical capacity to achieve that goal. Third, or fourth, that achieving the ambitious goals of the global technical strategy is in the path to elimination. Or in other words, if we don't first reduce disease and death in a dramatic way, as is proposed by the GTS, we will not eliminate we won't eradicate malaria. If we don't manage to eliminate malaria deaths, we shall not eradicate malaria. And therefore, a full support and moving forward with the GTS is on the path to elimination and eradication. In closing, elimination and eradication programs are laudable goals, but they carry with them an awesome responsibility. There is no room for failure. Careful and deliberate evaluation is a prerequisite before embarking on any such program. And worth reinstating once again that elimination and eradication are the ultimate goals of public health. The only question is whether these goals can be achieved today, or it will take us 10 or 15 or 20 years. But the goal must be very clear. As I was asked to speak of history, let me end up with a quote from 1939 by the malariologist Boyd, which I think does capture very well where we need to be and how do we approach this goal of eradication. Malaria control should not, in other words, read now, malaria eradication should not be a campaign. It should be a policy, a long-term program. It cannot be accomplished or maintained by spasmodic efforts. It requires the adoption of a practicable program, the reasonable continuity of which will be sustained for a long term of years. Thank you very much. Thank you.
Yes, a little. So, without calling upon you, if you have a seat next to you that is empty, I have 150 people standing for this entire thing. Please raise your hands. There are plenty of chair guys. Don't be shy, unless you love standing for an hour and a half, and there are people who do that. And I will give you 1 minute to wander up. There really are chairs up here, and we really want you to feel and enjoy this because it's going to get even more fun. Very good. There's at least 20 in the first 3 rows. For those of you that are truly not shy, you're malaria people. You can't be shy. Come on. Great. There's more up front. Keep on coming. Very good. Janice, come on. Keep on coming. I know you guys. I can start calling on names. Please. Okay, great. A little bit better. All right. My pleasure to introduce Dyann Wirth, Harvard T.H. Chan School of Public Health, to talk to us about the next step, which is, of course, expanding the malaria toolkit. Thank you.
Thank you very much, Gina and Chris, for the invitation to speak at this important and memorable honoring of Alan Magill and his vision. Chris and Regina asked me to talk about kind of this idea of using basic science to expand the toolbox. And so that's what I'm going to focus on, and I'm going to focus on this using a vision that I think Alan had in terms of how science can be applied. So in terms of progress in the last decade in basic science, we have made amazing progress, both in our understanding of the basic science of the organisms involved, the parasite, the mosquito, and the human host, and in the technologies that we can apply to address this problem. So there is enormous hope. And I will highlight a few of these. I can't do justice to everything, but this meeting is filled with new discoveries by many of our up-and-coming young researchers, and I encourage all of you to attend as many of those sessions as possible. Now, as Alan looked at this, and Bruno, in fact, used this a related slide, Alan really looked at elimination and eradication through an evolutionary lens. That is, the idea that as parasites interact with their two hosts, it's sort of an arms race. Everybody wants to survive, and everyone works to their advantage to live the best life, to be transmitted in the case of the parasite, to survive in the case of the mosquito, and to survive in the case of the human host. And so there's this constant pressure and evolution. And we're going to talk about that. We're going to use that lens to look at basic science discoveries. So Alan really wanted us to incorporate this thinking in everything we did in elimination and eradication. And I think that's very important for us to consider. Now, Bruno's already mentioned the two kind of prominent evolution or selective processes that we know about, and I'm going to talk in some detail about those and new approaches to how to overcome those. But then I'm going to move toward the middle and end of the talk to talk about evolutionary or selective pressures where they certainly exist, but we don't understand their origin. So as Bruno mentioned, and this is again a slide from Alan's deck that he used for his presidential address, that in fact emerging resistance is one of the biggest challenges and certainly one of the results of evolution. The two resistances are drug resistance. In this case, I'm going to talk about artemisinin resistance and insecticide resistance or pyrethroid resistance. I'm first going to turn to the insects. We know that not only this is one of the obstacles to eradication. We can see it coming. There's resistance, but in addition, the measures that we currently have to deal with insects, for the most part, only deal with the indoor biting mosquito. And so, in fact, anything else escapes control. And so one basic science strategy, which I think supersedes the use of insecticides or chemical approaches, is to actually genetically engineer the parasite. And over the last 3 or 4 years, this has become possible in a very facile way. And this is the process of genetic engineering in which we can see gene drive or genes or particular selectable genes moving through populations. And so I'm going to show you just one example of that. Here is the idea where you can actually modify the genome of the mosquito either by introducing something which could kill the parasite. So you would, and then replace the mosquito population containing this, with the mosquito population containing this gene, or alternatively, you could introduce sterility and suppress the population of mosquitoes. Both of these have been tried, and there's enormous progress. This field is moving quickly. There are several sessions here. And so the concept is that by engineering sort of a beginning mosquito, that this trait, every time the mosquito mates, all of the progeny will carry this trait, and eventually, over a relatively short period of time, the population is replaced. This has enormous possibilities, but of course, there are issues of ethical issues, technical issues. Is it going to work? But this gives us hope that this is a completely different way of approaching insects by engineering the insects themselves. Okay, now to drug resistance. We all know that drug resistance is a problem. It has been in the history of malaria and in the history of infectious diseases. Technological advances made the possibility of identifying the mechanism by which parasites became resistant to artemisinin going fast forward. From the discovery of slow-clearing parasites to the identification of one of the genes involved took only a few years because of high-tech approaches of cell culture and high-throughput sequencing, coupled with excellent field work. In the work of Arie et al. And Mioto et al., they were able to identify the gene that's associated with resistance, and in fact, and now there are surveillance programs to identify this and make sure, wherever possible, to slow its spread. However, resistance is an true fact. No matter what we do, the parasite's going to develop resistance. So in fact, the basic science approach is in fact to generate much more information, much more opportunity to develop new approaches to drug discovery. And there's been an international effort over the last now decade to screen millions of compounds for the ability to kill not only the blood stages of the parasite, but liver stages and transmission stages. And this, in fact, was one of Alan's sort of key visions, and you're going to hear a talk later in this session about one of those potential developments. But in fact, this phenotypic screening, this way of identifying compounds that kill the parasite, identifies lots of compounds. And this is from the work of the MD-TIP consortium led by Elizabeth Winsler at UCSD, in which a group of investigators have been screening, have been taking the compounds that were positive to kill the parasites and identifying the targets of those compounds to have a much better picture. This harnesses the technology of cell culture, high-throughput technologies, and high-throughput genomic sequencing, coupled with CRISPR or other genetic technology and crystallography to really begin to accelerate the discovery of new targets and potentially new drugs. I think Elizabeth's vision for this is that, in fact, the entire genome of the parasite will be annotated both for druggable targets and for many of the new resistance mechanisms which are appearing under this set of studies. Of course, this works, almost all of this work is focused on Plasmodium falciparum, but in fact, one of the biggest challenges to malaria eradication from a biological sense is the Plasmodium vivax hypnozoite. That parasite exists in the liver and remains dormant, and it's experimentally, until very recently, has been inaccessible for drug discovery or drug development or basic biology. But recent work in model systems, in humanized mouse systems by Stefan Kappa's group, and what I'm going to show you in the next slides in using bioengineering and livers on a chip by Sagita Bhatia's group, it's now possible for both of these groups have identified hypnozoites or small forms in vitro culture. And this really allows them to now begin to investigate potential drug targets, but also to investigate the biology and understand this critical obstacle to eradication. Okay, so now I'm going to step back and look at evolution a little bit more broadly. And one of the things that many of us who have been involved in using genome sequencing, it is really the enabling of genome sequencing to sequence now thousands of genomes of the parasite, that we're really beginning to understand the evolution of the parasite at its most fundamental level, at the level of DNA sequence. And I just show you a slide from one of our first papers in this area, in which we looked at all of the predicted genes of the parasite and asked how diverse are those genes. And most of them have very little diversity, but then there's this very long tail that shows very high diversity. That is, parasites from around the world or even from in a region are quite different from one another. And I've plotted here the approximate location of the various vaccine targets that were in development at this time in 2007. So one of the things we predicted, and Chris Plowe has been involved in this work for one of the antigens, is that in fact this data told us that pre-existing in the population of parasites in the world were parasites that were divergent, and perhaps if you vaccinated with a single allele, you would only see a protection against that allele or closely related alleles. And so, in fact, we were able to directly test this hypothesis in a collaboration with the large-scale RTSS vaccine. This work is published. I won't go into it in any great detail, except to say that we looked at every parasite escapee, every parasite that occurred either in the vaccinated or the control arm of the vaccination trial with the Broad Institute, using a technology developed for high throughput and multiplex sequencing. So we examined every parasite in those individuals. And together with Peter Gilbert's group, using analytical technology developed for HIV vaccines, we tested for a sieve effect, asking whether parasites that carried the same allele as the vaccine, the RTSS vaccine, were selected or removed from the population as shown in this kind of cartoon diagram, so that the red parasites, in fact, are eliminated from the population. And the data from that trial clearly showed a significant sieve effect for the homologous strain. And so again, this idea that diversity in the parasite population has enormous implications for vaccine implementation. So there are many strategies to get around this. You could use a strategy that's used in the area of flu vaccines, combining different allelic types, or in many bacterial vaccines, or an alternate strategy, which I really think wanted to highlight here, is to actually concentrate on some aspect of the parasite biology. I'm going to show you work from Julian Rayner's group in which he concentrated on molecules that are involved in the invasion of the merozoite into the red blood cell. He was able to identify through a sort of medium throughput technology a molecule that was invariant, the RH5 molecule. And when he raised antibody to this molecule, he could show that it cross-protected against many strains with from many different geographic origins. And this gene, in fact, is almost monomorphic, or it has limited diversity. So this is an alternate strategy. Pick a vaccine candidate that doesn't show diversity in natural populations. Okay, so the final evolutionary point I want to make is kind of related to what both Pedro and Bruno showed, this concept that we are really bending the curve. We are changing the kind of frequency of malaria parasites in the world. And that has some consequences. I mean, you can see it in these very nice heat maps from the Atlas project. You are seeing reduction in the intensity or the prevalence of malaria. But in fact, that is a biological process. And I use this diagram, which I took from a review article from Zhishen Zhu, written in 2003, because I think it captures in a simple way this concept of bottlenecking of populations. And this is a very important biological concept. And in fact, there have been various bottlenecks in the evolution of the malaria, this is falciparum, from its primate ancestors through to modern day parasites. And now we're introducing massive changes in the parasite population, putting stress on the population to reduce the numbers, and only the survivor parasites under these conditions are going to thrive. And of course, one way to test that is to see, can we in fact see evidence for this evolutionary bottleneck in the parasite populations. And this is work from my own lab in which we've been looking using genetic markers or whole genome sequencing under different conditions of malaria transmission, going from high transmission to low transmission. You see a reduction in the number of parasite genomes, and you see changes in the parasite genomes and their relatedness down to single or small numbers of populations. This data is again published, so I won't go over it in detail, but in fact, you can actually map, this is in Chess, Senegal, with our collaborator Douda and Dyann at Czech Antioch University, led by Sarah Volkman and Raisha Daniels. You can see actually by just plotting clonal parasites over time, over years, that in fact in the kind of middle 2000s, after introduction of RDTs and ITNs and IRS at high levels, you saw a bottlenecking and increase in clones, and then things perhaps fell off a little bit, but then reintroduction of these measures, and one can detect these signals with great precision using genetics. Now, in fact, so this technology is available. It allows us to track what's going on in detail. And there are several presentations during the meeting from many groups talking about these approaches. But it's interesting, as I was going through Alan's deck, in preparation for this, actually Alan sort of anticipated these results even before they existed in saying, if you had a marker where you could tag parasites, you could barcode them, you could identify, have a zip code for each and every parasite, you might be able to understand something about transmission and movement of parasites. And indeed, he laid that out in some detail in his thinking about how to approach eradication and with the evolutionary principles in mind. Well, in fact, from the UCSF group, from Brian Greenhouse, in fact, they're exactly applying these kinds of approaches to understand transmission networks in Swaziland. And so I think, again, I want to end now with just probably the final tool that's the most important one for elimination and the eradication. It's not really a new tool, but it's a tool we need to use every day, and that is the educational tool, the sharing of information. And this is a picture of Alan teaching in the Science of Eradication course only a few months before he passed away, where he was sharing his vision as he had every year. This is a course that Pedro, IS Global, and Marcel Tanner from Swiss TPH and my group at Harvard have run now for the past 5 years. And Alan was an active participant in that course, helping us shape what kinds of information the leaders that were going to take the baton, take it to the next step, needed to have. And that is probably among the most important legacies. And I just end where Pedro and Bruno started, with the stamps commemorating the eradication of malaria. This is a humbling slide. These stamps were printed to celebrate malaria elimination in the previous campaign. With that, I'll end. Thank you.
Thanks, Dyann. And I have two announcements as our next speaker comes up, Chris Drakeley from the London School of Hygiene and Tropical Medicine, to talk about malaria's footsteps. The announcements are, number one, I should have mentioned at the beginning, this session is being webcast, so you can watch it live as we go, and also you can catch it in perpetuity afterwards. I hope the speakers are all okay with that. Sorry for telling you after you gave your talks. And secondly, many of you know that Alan Magill used to carry a malaria coin, originally created by the malaria vaccine program at the Walter Reed Army Institute of Research. On it, it said, Malaria delenda est, after Carthago delenda est, from Cato the Elder, a Roman senator a couple hundred years BC. It means malaria must be destroyed. I've got mine too, in Latin. And I understand that a few days after he died, several of his colleagues went out and got that tattooed on themselves. And so I just wanted to show you this. And I had actually requested that we have a tattoo artist here at the meeting for anybody who wanted one. But in lieu of that, these temporary tattoos, which I have, are available for $1 each outside. I have a small personal supply, so you can get them outside this session or at the registration desk. So with that, Chris, please come to your talk.
Okay, thanks. Afternoon, everybody. Thanks to Gina and Chris for inviting me to what is a fantastic symposium. I'm really delighted to be here. I was to be a co-chair with Alan on part of the malaria series that Bruno alluded to earlier, and we'd had a really lively conversation about the concepts of what would comprise that document, and I have to say it was made much harder by his passing. And I'm sure he would have made an excellent job of it. It should be out soon, and so you'll see the fruits of that work. So I'm going to follow on from Dyann, really, and talk about how we can use how our body reacts to that diverse array of parasites that's exposed to look at malaria transmission dynamics, to increase our chances of identifying where transmission is occurring, to better target control and elimination activities. And the unique facet of antibodies is that they last for much longer than the infection in the mosquito or the human. And so what I'm going to try and do, the outline, is just to try and take you through why serology might be useful, and then to go through some of the developments that we've had recently, and to highlight the challenges of an adoption of any new technology. So we're going to start, why is it useful? So as I said, the infection in the mosquito is a matter of potentially days or not very much longer than weeks. In humans, I think there's some debate about how long infections will last depending on the level of immunity, but it's certainly not as long as the immune response that infection will generate in the human host. And we've had some various estimates of that from years to... Oh, I'm not getting the hang of that. Various estimates of that from 5 to 40 years, depending. You can look at individuals who've migrated to non-endemic countries and still find antibody responses. What you will see is that the levels of antibody responses drop over time, and that becomes relevant, and I'll talk a little bit more about that. So practically, what does that allow you to do? So in that schema here, what I've got are a series of parasite surveys in this. This is a small population, but you can scale it as much as you like, and they can be as frequent as you like. I'm talking about potentially surveys every 2 months, every half year. And this is what we see with our parasite infections. We can see that 3 of those surveys might be negative. If we were looking for malaria parasites, we might assume that there was no infection in that population. But if we did a serological survey, what we would expect is to see the footprints of that infection within that population here, and with some nod to the amount of detection we would have since the time since that infection originally took place. So that infection was that long ago, would have the lowest level of antibodies. So as its very simplest, what this approach allows is the detection of many more infection events or exposure events than simply a single time point parasitological survey. So when we're looking for these increasingly rare, sporadic infections, the idea of having a more sensitive diagnostic that would be an additional tool, was not proposing it to be the only tool, but as a tool in your toolbox, to be able to use that to detect exposure events is really potentially very useful. And so what would that look like if I now try and look that over time? This is some work that we've done with Edward Wenger at IDM in Seattle, thinking about how we might perceive the use of a serological test in an endemic population. And what you can see, probably just make out in the bottom here, is what we have is our RDT with relatively low detection threshold, PCR with a little bit more, but then we're looking at antibody levels associated with different time periods of antibodies with different half-lives, essentially, with a longer half-life of, say, something like 18 months in the light green here. So already you can begin to see how much more transmission or exposure we would detect by looking at by incorporating some sort of antibody into our survey methodology. As there's been quite a lot of... The way that we interpret serological data here is simply we can either look at a seroprevalence measure, so the proportion or percentage of individuals that have antibodies, or typically as exposure to the parasite is related to age, we can look at fitting a simple line through that age seroprevalence, which gives us a seroconversion rate, i.e. that's the rate of individuals which become antibody positive over the course of typically a year. And what we've shown is that essentially that seroconversion rate, which is the blue triangles there, is much more tightly correlated with the entomological inoculation rate than the paired parasite rate derived from those same samples. So it's a much more consistent measure of transmission than parasite rate. As I showed you in the previous slide, parasite rates can vary depending on when you go. Seasonality could be just after drug treatment. So there's good provenance there for this as a method. And then just trying to give you a sense of the scale, I borrowed this from another one of the Malaria outputs on interventions and modelling, looking at the range of transmission which we might expect and the other different markers or measures of transmission that we might commonly encounter, such as EIR, parasite rate, weekly cases and annual parasite index. And again, these are predicted seroprevalence and seroconversion rates. The thing that is evident here is that at this very, very low transmission level where we are finding it difficult to measure and parameterize the conventional measures, serological measures are actually giving us quite a clear indication, or we can begin to still grade transmission intensity within this very low range. But it's not new. It's been done before. There's a nod to the history that we heard from Pedro and Bruno. Here are two sort of classic examples of using seroprevalence before. This is Mauritius, the elimination on Mauritius in 1973 by Bruce Schwott. A lot of students will have read his classic textbook. And this is looking at seroprevalence 5 years after eradication on Mauritius, or elimination I should say. And what they then there is showing, as you would expect, in children born since transmission has ceased, no evidence of exposure, no antibodies. So what we have here is this unique facet of malaria, of serology, in the sense that it recreates the exposure history over time, and we can begin to look at changes in transmission intensity. And here what he did rather simply, probably just as good as some of the models that we've done, is just simply fit regression models to get an idea of how the transmission intensity at different age groups and different parts of time. Here is the classic Garkey study, looking at this kind of filled area is the antibody levels, so these are levels rather than prevalence, in the control groups, and the lines going down are in the several weeks following intervention. So what you can see is population antibody levels decreasing over time, and then when they stopped the intervention, population antibody levels returning back up to pre-intervention levels as transmission is re-established. So there's a strong historical precedence, and when I first started to talk to Alan about serology, he said, well, why don't you just do that? They've done it before and it works. And so he was supportive but not necessarily effusive about how modern technology might adapt to this. And so his first suggestion was that we try to see whether commercial kits that are available for screening blood transfusion, certainly we use them in the UK and other European countries use those, to see whether they could be adapted for epidemiological purposes. And we're in the process of doing that, but whilst it's a very important piece of work, it's actually quite boring. So I'm not going to show you any of that. But he was always challenging us to say, well, okay, if this isn't good, and Dyann gave a very good kind of idea as to why it might be the variation in parasite development and also our inherent human genetic variation as varying responses, then what would be the new platforms that might take this forward? So I've collated some information from various people who are working in the area now, just to show you kind of how we're moving forward with the various approaches to generating serological data. So the whole idea of the previous historical work, what they used was essentially homogenized parasites. And what you have in a homogenized parasite, obviously, all the antigens that the parasite expresses, and not only that, but they are in the right relative amounts to each other. However, they're typically only one strain of parasite, and it's not so easy to produce vast quantities of those antigens in a standardized way. So some work kind of initiated and led by Phil Felger at UC Irvine began to look at, well, let's look at all the malaria parasite proteins and see how serology can be used to identify and to hone down important antigens that might be used for, obviously serological markers of exposure, but also various other approaches such as vaccine development. So what Phil did was essentially look at all 7,000 odd proteins of malaria parasites and examine antibody responses to all of those. And what you can see is that actually relatively small proportion of those actually are responded to by the human sera. So that's actually quite good because it allows us to hone down on immunogenic antigens, ones that might be used in serological assays. You can see here his panel of naive US adults and then some naturally exposed or some sera from naturally exposed Africans here, lighting up his microarray panel. So what we've got are at least a reduced range, but still a range of antigenic targets that we might incorporate. And Phil and his colleagues at UC Irving, together with many investigators in the NIH-funded ICEMA program, are now beginning to examine antibody responses to this kind of honed-down list of antigens as part of a multi-site survey. Here he's shared some data with us from 3 different areas of Zambia, looking at differential responses to the top 30 antigens between children and adults in varying areas of varying transmission levels. And you can quite clearly see that not only does the response scale with the amount of malaria there is, but it's also lower in children and adults, suggesting that it's on the right track. Chris's group have taken that further. So, as Dyann described, the highly variant antigens, multiple different clones expressing variant antigens, or antigenic variants in this case, of some key Merozoite stage antigens. So, Chris, together with Phil, has developed a panel or a microarray of variant AMA and variant MSP1 antigens. And the relatively simple take-home there is that adults react to more variants, i.e. they see more clones in their lifetime than children, but the number of variants that children recognise increases after the transmission season, which is again something that you would expect to see. But the relevance there is that actually we can begin to potentially really finely dissect the response to not only have an underlying molecular kind of characterisation of the parasites, but use that to overlap with exposure to those parasites in the population. So Chris's group have taken this even further and drilled down a lot more. So the previous array was based on proteins. Here what he's done is an array based on overlapping peptides. Some of us will remember the old peptide array PEP scans that we used to do on ELISA plates, very time consuming. Now the microarray technology has completely revolutionised our ability to do that, to look at that in depth and in large numbers. But the novel element here is that what is also on the array are peptides from Anopheles saliva antigens. So obviously when a mosquito bites, it inoculates saliva as an anticoagulant, and you as an individual will develop an immune and antibody response to that saliva. So potential marker for exposure to bites. So again, this is a really neat way of drilling down onto identifying markers of exposure to mosquito bites. And the panels show antigens from different Anopheles species and their relative seroreactivity in North American adults, Southeast Asian adults, and Malians. And the Southeast Asians are clinical cases, I believe. How we then use those in an epidemiological context is potentially where we're really interested. So whilst we have the technological laboratory-based development, what we have is a desire to deploy and test those tools epidemiologically. And this is data from Vincent Corbel and work on the Thai-Myanmar border, showing essentially heat maps of high antibody level responses to Anopheles proteins, correlating with the red dots, which are high levels of anopheline catches and EIR. So again, the potential to use that as predictive to where the bites might be happening and to target your interventions accordingly. So I talked a little bit about the beginning about the identification of antigens of recent exposure. One of the issues with serology is that, or antibody responses, it does last for a long time. What we've been interested in, together with colleagues at UCSF, is identifying individuals or identifying antigens that are associated with short-term antibody responses. So in this bottom left panel here, what we have are a single individual time since exposure, all the antigens there measured against, or that's one antigen against a series of individuals, looking at the decline since the time from infection. And there we're identifying all the antigens which show that same profile, a decrease since the time since exposure. So when we begin to try and predict time since exposure or incidence of infection, if we use 600 antigens, that prediction is not very tight. But if we use the top 10 antigens from this particular plot here, we can find that this is time since exposure, so a rapid reduction in antibody levels. This is incidence, an increase in responses to those antigens. So what we've done in London is make those antigens. We've actually produced them and we've put them onto the Luminex platform and the bead array platform to look at kinetics post-exposure in a very detailed cohort that our colleagues at IDRC have been managing together with UCSF. And here are individual plots, and you can probably just make out the key at the bottom there, a malaria infection, and what we can see are select antigens dropping off since the time since exposure. And when we combine those antigenic responses, what we can see is here are some of the top 10 hits of the ones we've managed to actually produce recombinantly, dropping quite rapidly after time since they were infected, whereas some of the more classic antigens show a much shallower drop in antibody level. So Pedro talked about Plasmodium knowlesi, and I think that we're kind of interested in understanding the response to other species because we want to have a fidelity around what we understand our response to falciparum. And also in terms of knowlesi, it's a relatively rare event, and it allows us a kind of a biological attempt to look for rare events within a population. So what we've done here is the opposite. We've tried to identify antigens which have no sequence homology with Plasmodium vivax, for example, because there's quite a lot of sequence homology, but also antigens which exhibit heightened immunogenicity, i.e. are likely to induce an antibody response. So that's based on hydrophobicity, it's based on whether they're membrane bound, etc. So a series of qualities that define immunogenicity. We then produce those in a recombinant way and put them into ELISA in a population survey from Malaysia. This is our falciparum plot, which shows very, very little falciparum exposure. As Pedro said, Malaysia is quite clearly on the way to elimination, but there was some historical exposure. This is the knowlesi plot, nothing like a pattern that we could observe, suggesting that transmission is very, very different, and that's what we know about Plasmodium knowlesi. The risk factors are not specifically age-based, but they are spatially. So when we put those on a map, this is that particular plot. This is Borneo, Malaysian Borneo, and this is the district of Kudat. What we can see for Plasmodium knowlesi is that the antibody responses tend to be on the outside of the village, i.e. in closer proximity to forest, and the falciparum responses that exist tend to be peridomestic. So also advances, how do we use this in terms of an analytical sense? And this is just a quick flash through some methodologies that allow a sample size to be developed if you wanted to use the assay as Alan had originally suggested it for elimination, certification of elimination. And here, a different approach for freedom from infection, where we're thinking about actually the probability that disease does not exist. And Gillian Stressman has a talk on this. This is something that we've been developing. So finally, one of the main things that we can do with the serological approach, simply because you do get many more seropositives, is develop a mapping approach. And this is some recent, definitely hot off the press data from Myanmar, the recent survey conducted by Malaria Consortium, DMR, in Myanmar. This is the original Malaria Atlas Project map, which is not particularly informative, or primarily based on the lack of information going into it. This is the annual parasite index map that is used to define the surveying structure, the cluster survey. And this is the serological map that has been developed with collaborators Pete Gething and Ewan Cameron in Oxford. And what you can see immediately is a lot more granularity. It maps it traces the API very well, but it also provides a lot more granularity in the mapping. So that's something that I think offers real possibilities. So the challenges, I've got one slide, but this is really where most of the endeavours need to go. What I've described is a variety of platforms providing a massive amount of data. And our challenge is to translate those multiple antigen responses across the top from multiple people into a series of, well, yes or no, is that positive, is that not? And then to say, well, okay, we've got multiple antigens, what do those antigenic responses mean in terms of historical exposure or recent exposure? And then actually, how does that fit when this population come from one particular area? How am I going to decide what that antigenic response means, the seroprevalence here, for these particular areas? And how does that translate to a village layout or a region layout in order to specifically guide some sort of control or intervention procedure? And that's really where the challenge is at the moment. And I remember discussing this with Alan some months before he passed away. And he was saying, well, it looks very promising, but it is slightly aspirational at the moment. And I kind of reminded of him that a little bit of aspiration wasn't a bad thing. So I think that my summary would be that the antibodies has an excellent potential as additional measure of transmission, particularly in low transmission settings, with that unique facet of being able to look back in time, which none of the other metrics that we have are able to do. But the major issue is providing a standardised assay with clear use scenarios. And then I'd just like to thank a few people that have provided data and helped along the way. So thank you.
Thanks, Chris. And our last talk is the appropriately titled Knockout Punch by Thierry Diagana from the Novartis Institute for Tropical Diseases in Singapore. Thierry.
Thank you. Thank you, Gina and Chris, for the invitation. It's a great honor to be speaking at this symposium. I haven't had the chance, actually, to work a lot with Alan. And unfortunately, most of my interactions started when he was actually appointed at the Gates Foundation. But all I can say is that his commitment and passion to exterminate malaria with extreme prejudice is how he usually described this. Was just infectious, and I was one infected, so I'll get the tattoo after the session. So I don't know how this works. The green arrow is forward. Okay, so my first slide is appropriately titled Malaria de l'Andes, which is the purpose of the tattoo. I'm not going to spend too much time for this audience. Everybody knows the tremendous accomplishments so far in shrinking the map of malaria and getting it to where it is today, which is tantalizingly close to eliminations. But the difficult news is that probably that final mile is going to be a lot harder, and Pedro already alluded to this, and some of the challenge that we'll have to try to really put the final knockout punch. And for that, we will need new tools. And this is what I think I will be giving you a little bit of a flavor from a drug discovery perspective of where we've been this past 10 years and where we hope to be for the next decade in drug discovery. Before I do that, though, I just want to reiterate something that's actually been said already, which is the threat of artemisinin resistance. Resistance is inevitable. I think Dyann said that earlier. This is just a fact. It will happen to every single one of the drugs that we have made and that we will make. And therefore, we need to continue to make more drugs. And I think that's a case that needs to be repeated because the successes of the past decade sometimes kind of diminish the urgency for us to make new drugs. And I think lately, with the discovery of piperacin losing its efficacy in the GMS region as a result of artemisinin resistance spreading, is another example of why it's so important that we have new drugs. So the target product profile of SIRCAP, single exposure radical cure and prophylaxis, which was laid out about 9 years ago by the Malaria Consortium, actually has the aspirational vision to deliver a drug with a single dose that can treat, prevent, and block the transmission of the disease. Obviously with an objective of improving patient compliance, but also the potential to drive mass drug administrations and eliminations. From a drug discovery perspective, some of the challenges, and there are many, but a few that I've highlighted here. One is that it's very unlikely to come from a single molecule. Single dose with a single drug is not going to be achievable. It's going to have to be a combination, which really highlight against the case for multiple drugs. We need really to have a panoply of drugs with multiple profiles to meet that profile. And the other challenge is that we have to keep in mind that, as much as an aspiration that single dose is, it's likely that we will get there through an incremental improvement on the standard of care. And that the silver bullet, which will deliver in combination single dose cure, is probably going to be an iterative and incremental process which will require multiple products before we actually get there. So, prevent, treat, block, I think it's been highlighted in the multiple talks before me, so I'm not going to spend much time. Just to say that all drugs treat. Every single antimalarial drug treats, and that's normal. It has to be because that's where the most pressing medical need is. But not all drugs prevent, actually very few do, and even less drugs actually block transmission. In fact, we have only one that has documented transmission blocking potential. So I think the vision to deliver that single dose cure will have to include these two aspects, and the good news is that there are progress being made in that area in terms of high throughput screening, both on the liver stage and the gamidocyte, which should enable to identify those type of molecules. So that has the points that I've made basically have been captured in what the Medicine for Malaria Venture have put together as far as target candidate profiles. There are 4. TCP1, which is a fast-acting, highly active compound that can very rapidly decrease the parasitemia in the patient. TCP2, which has a long half-life and can basically mop out the parasites that are left from this TCP1 compound. Then TCP3, which targets the non-replicating form of the parasite, namely the hypnozoite of Vivax, and I'll say a few words at the end of the talk, and gamidocyte, the sexual stages of the parasite, and then finally the TCP4, which is the chemoprophylactic profile, which targets the liver stages. So we need to get drugs that meet all of those profiles, and there's been about a decade now of work that has gone into identify such molecules, and as I said, and as I will show you in a minute, it's been quite successful. But the real drivers behind that, and here I have to give credit to my colleagues at GNF. Elizabeth Winsler, when she was actually at GNF, was one of the pioneers that leveraged the really tremendous progress in automations that our industry has seen, which really truly enable millions of compounds. And in fact, I can't keep track of this. Every time I talk to MMVs, there are 6, 7, 8, 9, 10 million compounds that have been screened. This is really an unprecedented effort that's gone, where really we now have thousands and thousands of potential chemical starting points, which really have truly been enabled by that approach of phenotypic screening. But it's not only giving you a fast route to actual drug candidates, it also gives you the opportunity to identify new vulnerability in the parasite and new drug targets that actually were not characterized before. And we've had, with the advance of chemical biology and drug resistance studies, really shown that many of these compounds can be mechanistically deconvoluted, and we can actually discover their target as well after identifying the hits. So using that approach, our consortiums and many collaborators and many people, some of them are here, working on these two compounds, KE609 and KF156, we've been able in 6 years to deliver to proof of concept in patients from screening to proof of concept, which by all accounts in the industry standards are actually quite fast timelines. These compounds all the POCs have been published, so I won't talk much about it, except to say that 609 and 156 both have quite rapid parasitemia clearance time, with about 12 hours half PCT for 609 and 48 hours, less than 48 hours for 156. From a global perspective, though, if you actually look at the pipeline and this is the latest pipeline from the Medicine for Malaria venture, you can see that across the board, from research all the way to access, you have quite a rich pipeline with a lot of molecules that actually have come forward. One thing that's worth noting, though, is that for the most part, they all come from phenotypic screening and HTS. So that slide that I've shown you with these robots at work actually have translated in an unprecedented level of new molecular entities with new mechanism of actions that populates this pipeline. The second point that's interesting to note is that actually there are also mechanisms that are very common, and we have been fortunate, and I'm going to give you a little bit more about this example of PFATP4 to identify the target of one of these molecules. But at least 3 groups independently in another independent screening identify structurally different compounds that actually also act through this. And I think I just want to say, I'm going to talk about PFATP4 in a second, but this is not true just for PFATP4. We actually have in this slide here shown, you have this compound and the 390048, which actually acts through PI4K, and we have many compounds in our decks and our hits that are actually acting through that mechanism. Same with DHODH. DHODH is a very privileged target for malaria, and we have multiple series that are coming at our screen that are coming through DHODH. And I can also quote another example of the many DHFR and the cytochrome BC1 hits that we found in our library. So I think at the end, when we'll be going through these screens, we'll have to ask ourselves the questions of really how many truly unique mechanisms we have in these sets, and probably challenge ourselves to screen differently in order to discover additional mechanisms. So PFATP4, as I said, is a very promiscuous target because we have at least 4 chemical separate classes that act through this gene. It's a P-type non-circa ATPase, which was predicted to actually be a channel for calcium, and it turns out that it was not true. And I'll say you a few things in a second, but it's depicted here with the P domain and the 3 transmembrane domain that you can see there. And in a paper that we published in a collaboration with Elizabeth Winsler at GNF, when she was at the Scripps actually, we were able to show that some mutations, single nucleotide polymorphism in that gene, were actually conferring drug resistance to K609. We furthered this work with David Fiddock and actually identify additional mutations. All of them are highlighted here in red, and many different groups using some of those compounds have also identified other mutations that are also shown here. What was interesting is that, sorry, if I want to go back, it's this one. What was interesting is that some of those mutations actually give cross-resistance across these different classes of compounds. Karen Kirk at the Australian National University nicely shown that actually that gene was actually regulating sodium, and that by inhibiting that pump, we had an intracellular accumulation of sodium, which turns out to be toxic to the parasite and led to this very rapid clearance time in patients that are shown here in Vivax patients and falciparum patients, where you can see that within 12 hours, half of the parasitemia is gone. And one thing that was interesting is it's a work that you can't see here because the slide is not working, but what you should see is non-infected parasite, non-treated infected red blood cells that goes through these slits very quickly and very easily, but compounds that when we treat with the compound K609, the rheology of the red blood cells is actually changed, and the compounds actually induce a lot of rigidity in the red blood cells, which cause those cells to get stuck. And we believe that this additional property of the compound enhance the spleen clearance, which actually helps with those very rapid clearance times. So it's a very unique mechanism, which is nicely represented in the portfolio, which I think will lead to a compound with clearance time that actually faster than artemisinin, which is obviously quite nice to have. So I've shown you that things are well, and we can definitely all agree that within the pipeline, there is a lot of reason to hope. But like many of our colleagues are saying, the glass is half full, and it's not going to be done until it's done. And so what are the challenges to get us to where we need to be in order to really complete the malaria elimination agenda? So I've highlighted 3 challenges that I can think of. There are many more, but I'll just say a few things about first, the need to really improve the way we design combination therapy, the need for better safety profile for our therapies and how we could potentially get there. And then I'll finish with a few words on Vivax and the particular challenge of Vivax. So combination, again, I'm not going to speak to this audience. Everybody knows why it's very important to do drug combinations. The rational shorter treatment, we can basically manage drug resistance and we can improve safety margin using that. This is pretty much expected now by the WHO and by most of the global health agency that for infectious disease management, we need to have combination therapies. It's true for HIV, it's true for TB, it's true for malaria. And then we have a regulatory landscape which is evolving. There's a new FDA guidance which actually somewhat relaxed the need to demonstrate efficacy of the individual components of the therapies if you can show major advantage on your actual combination products, which are all opportunities we should leverage. The challenge that we have is that today's combinations are mostly empirical. We're basically dealing with a set of old drug partners, which is actually shrinking because we're losing them to drug resistance like piperacin, but we still have, for example, lumefantrine and others. And the new drug compounds, actually, their safety profile is still poorly understood, so it presents some challenges in understanding the safety of your combination partners. We're mostly doing fast-acting and short half-life with a slow-acting, long half-life partner. Is this really by design, or is just because this is what we had and the best we could do at the time? And resistance to drug resistance is actually highly variable. If you look at every single entities, you have the atovaquone, where resistance is relatively easy to get, to chloroquine and artemisinin, which took quite a bit of time before we actually got really worldwide drug resistance. So what are we aiming for tomorrow's combinations? What I wish we could get to is actually rational design of this combination. So can we do matching pharmacokinetics? Is there an opportunity if you use certain mechanism of action to actually have 2 or 3 compounds that have matching PK and deliver what you need for single dose? I mean, those are the questions that we can ask ourselves now that we have really modern tools to understand pharmacokinetics and pharmacodynamics. Mechanistically, can we actually use pathways that actually synergize with one another so we can lower the dose and enhance the safety margin? I think there's some work that was presented also this morning earlier from Dyann Wirth's lab on DHODH. Can we leverage evolutions and put pressure on the parasite to contain the risk of drug resistance? So I think there's a lot of work to do in that area, and the good news is that we have a lot of tools to do better work and design our combination in a more rational way. The other challenge is safety, and that's driven by something that Alan actually was a big vocal proponent of this, is that most of the burden we're dealing with is actually you don't see it, you never see it, it's always under the water. And we're going to have to deal with this if we want to eliminate malaria. And the problem is that most of these patients actually do not seek care because they're totally fine, they're symptomatic by definition, and their parasitemia is probably not detectable by most standard tests. So how do we do to deal with this? Well, probably the only way you can deal with that population, is through mass drug administration. And that will require a level of safety which is even higher than what you need for a normal drug. And the challenge we have here, again, is that we're dealing with with drugs, current drugs, most of them have some safety challenges associated with it. And it's been fine the way they were used, in a properly medically supervised environment that present no challenges. But that's probably not acceptable if we're going to do this in a mass drug administration setting. And again, we have the challenge that the new drugs actually are not really well defined. So understanding our populations and being able to distinguish true drug signals from symptoms that are associated with malaria is going to be very important as we're characterizing those molecules, because we really truly want to understand the safety profile of a compound in great depth. And I think there's a lot of effort in defining the normal range of this type of event in the population in which we're going to test those new molecules is very important. And then, this is something more for us, but I think we all can accept the fact that low dose is always better. This is just a basic pharmacological principle that trying to aim for compounds that you can administer at low dose and maintain efficacy is definitely one way possible to de-risk safety. I'll finish with 2 minutes on Vivax. Vivax was something that Alan was just absolutely crazy about, and he used to say that the last soldier standing is probably going to be something like this, the hypnozoite. And that unless we know how to deal with this and do better than Primaquine, it's going to be very hard to envision malaria. So I'll finish with that last slide where basically, and again, Alan used to say it's music to my ears to hear that you're working on cynomolgi, because that's really a model that he's always viewed as a very good surrogate for relapsing malaria. It's a model which is very reliable and accessible because we're depending on monkeys and not patients, so we can actually control the type of infection. It's a model that relapses. If you look at the Leon Schmidt seminal papers, he followed these monkeys until 780 days. It's a model that we now can cultivate in vitro. In fact, in our lab now, we're able to maintain blood stage culture of cynomolgies for 200 days and observe gametocytes, which suggests that we can potentially transmit and bypass entirely the monkey. We can also cultivate it in vitro and have all the stages of the liver stage development, including the hypnozoite, the early schizones developing, and the more mature schizones. So I really believe that investment in that model will pay off because it's also a very good animal model for efficacy. So I'll finish in summary by just saying that the last decade has been extremely productive. We've seen novel clinically and chemically validated targets which can be leveraged. We have novel HTS assay across the life cycle on the blood, on the liver, and on the gametocyte. We have extremely robust and predictive model, and that's a luxury that many infectious diseases don't have, and that shouldn't be underestimated. It's really a tremendous advantage that we have over most infectious disease. And then finally, with the advance of the new compounds, we now have access to really an extensive preclinical, clinical PK/PD data set, which can and should be leveraged in order to better profile the compounds that are coming forward in the pipeline so we can understand and aim to rationally design combination therapy. So this is the work of a lot of people, and I try to give credit to some of them, but I'm just thanking them here in this last slide, and thank you for your attention.
Come on up. I have a question for the panel, so I'd invite you to come up. But we only have 5 minutes left for those of you that are running out. So let me tell you where we've been. First of all, while you were sitting here, the announcement of the Alan Magill Fellowship is in your inbox. And I think I ask that all of you look at it, not only for those that are eligible, for those that are not at the meeting and should be thinking about this. I think this is an incredible opportunity. Number two, as I was thinking about this incredible grant that the Gates Foundation has made, it's a long-term commitment, 20 years, wow, to have the kind of discussion. This was the easy one. These are the first. Now it needs to get even more interesting, right? But it points to the role of the society and the role of this symposium in coalescing the progress, the advances, the challenges that we're going to be facing. So the question I have for you, because we don't have time for a question and answer session, you can find these guys afterwards, is when we think about the challenge that Pedro raised, which is we need to be not program implementers but problem solvers. I would like each of you to identify one problem that this group and their collaborators and the teams should be solving and be thinking about the next year. I'm going to start with Bruno. No more than 60 seconds, please.
Okay. So, in Pedro's talk, he said, can we today completely eliminate malaria with the current tools? And I think the answer is actually yes, but it's incredibly hard and incredibly expensive and operationally very, very, very difficult. And that's mainly the case because whatever we do, we need to keep repeating it. And so my challenge to all of you is, it's one word, duration. So what if we have a vector control tool that you put out there or you apply and you only have to do it once every 5 years instead of a bednet once every 3 or IRS every 6 months or 9 months? What if we have a prophylactic drug that lasts, let's say, a full malaria season instead of a few days or a few weeks? What if we have a vaccine that actually gives you long duration of protection and not just high efficacy? And so my challenge to all of you is duration.
Thank you. Let's go down to the end. I know you've been working on drugs, Thierry, but you don't have to talk about drugs here. One problem that needs to be solved.
Well, I mean, it's actually Vivax.
Vivax.
I think that's the obvious one.
Janice likes that one.
We're still at a stage where we cannot screen, and I just mentioned the tremendous wealth that was generated by the incredible effort over the past 10 years of 6 to 8 or 10, I don't know how many million compounds that have been screened, but that alone is a true enabler. And we're nowhere close to this in Vivax.
Right.
And we need to be there. If we want to sustain the type of productivity that we want, we have to be there. So to me, that's a simple.
Chris Drakeley, one problem.
Yeah, I suspect that nearly 90% of the people in this room have contributed to the literature about asymptomatic or sub-patent carriage, and if we see another iceberg, we might drink ourselves to the grave. The question is how much do those low-density infections matter for transmission? Pedro talked about the fact that we have eliminated in a number of different settings without specifically targeting those individuals, and it's a much better attempt to try and understand where and in what settings and what malaria ecologies those asymptomatic carriers perpetuate transmission.
Asymptomatic pool. Dyann? There's one over here.
Well, Manoj is going to like this. I actually think that we need to revisit a vaccine. I think that if you think about the diseases that have been eliminated, they are, with the exception of guinea worm, they are all vaccine. Vaccine is a key component. And although there's a challenge, this antigenic diversity represents a challenge, escape from the immune system represents a challenge, I think it's time to really take the same kind of generalized effort that we have with drugs to identify vaccines and instead of focusing on a small number of targets that have been with us for half a century or close to it, let's try to get some new molecules and really test the envelope on the feasibility of a vaccine, because it has the advantage of duration.
Great. Pedro. We've got proponents on that one.
So, endorsing all the comments that have been made by the previous speakers, and moving perhaps outside the field of strictly sensu stricto.
I think you need to get closer to the microphone.
Moving beyond the research field by and large, malaria is not eliminated anywhere without real ownership by the country. So unless we understand what are the determinants whereby a country, a government, a society takes real ownership of the problem and they decide that they want to eliminate malaria and they put the resources or a very significant proportion of the resources that are needed, malaria does not get eliminated anywhere, irrespective of what any of us could actually do. So understanding better and working towards supporting the countries and the determinants that make that country take ownership of their own problem, I think will be critical moving forward.
Chris. You're sitting there.
Integration. It doesn't escape me that this panel up here is all people who work in the US, Europe, or Singapore. And that's partly because the focus of this first symposium was more on the upstream research, new tool side of things. And next time we're likely to be more downstream, kind of boots on the ground folks. But the program people, the researchers, the advocacy folks do tend to operate in our own worlds and speak our own languages. And for this to really succeed, we're really going to have to understand each other, communicate with each other, make sure that the tools we're developing are actually the ones that are needed and can be taken up by the people at the other end of the spectrum. So integration. So
there you have it. And the reason I did that is because there's two things you can do. One is actually fun, and you're trained to do that. You're scientists, which is answer the question. But the other one that goes with it is to know what question to ask. And that comes both from the science and both from the field and interaction and is critical to driving and engaging that next generation. We've had quite a road trip. We started with the case. We went to the strategy. We talked about the scientific base behind the tools, how to measure where we've been, because if not, like a previous boss of mine said, you're just practicing, and then the knockout punch. But really, the answer is you, right? It's engagement of you and what you represent as a scientific community with urgency. I won't tell you how many children died the 2 hours that we've been here. With commitment to the long term, because that's what it's going to take, and not because we're going to have the perfect toolbox today or tomorrow, frankly, but because we're going to be back hammering at this annually until it's done. So thank you. I am sure there's a drink in your future. Thank you very much for engaging.